Hello, this is Gina Piscatelli with the third lecture on the urinary system in A&P2 at Madison Area Technical College. This lecture will focus on only one function of the nephrons in the kidneys, and that is filtration that happens at the glomerular capillaries into Bowman's capsule. So before we begin talking about glomerular filtration, let's review the components of the filtration membrane. First, we have the wall of the glomerular capillaries, which contain fenestrations or holes. I think the size is about 8 nanometers. Then there's a basement membrane. That's the second component. 1, 2. And in here we have a network of kind of collagenous sort of proteins. And then we have the third layer, which is the visceral layer of Bowman's capsule. And there are slits between cells here called filtration slits. So the process of moving from the blood into Bowman's capsule is called filtration. And it's generally considered a passive process because it doesn't require ATP. It relies on fluid pressures and osmotic pressures, not necessarily diffusion gradients, but just general concentration versus dilution. And what gets across depends on the filtration membrane's permeability. That is, size and charge. Size of these fenestrations, charge in the basement membrane, which is negative, and the size of these filtration slits. So what we find is that blood cells are too large to pass through the capillary fenestrations, and large molecules such as plasma proteins and hormones bound to plasma proteins are too large to cross through the filtration slits. Also, that basement membrane being negatively charged will repel anything that's negatively charged, specifically large things that are negatively charged, like plasma proteins specifically. So they'll be repelled and they'll have to stay in the blood. So substances that are filtered out and make it into Bowman's capsule, which is kind of shown over here in this box. This is Bowman's capsule space right here, capsular space. Substances that are filtered out of the blood and into that space include water, ions, combination of ions that we call electrolytes, some nitrogenous waste such as urea, and then some organic nutrients, very small organic nutrients like glucose, fatty acids, and amino acids. So if we were to compare the filtrate that's in Bowman's capsule with what's in blood, we would see that they're similar, except, of course, filtrate does not have plasma proteins, hormones, generally speaking, or blood cells, right? Now, glomerular filtration happens at a certain rate, depending largely on blood flow. So we use this term glomerular filtration rate to kind of judge how well a kidney is filtering the blood. So to define glomerular filtration rate, or GFR, it is the volume of filtrate formed each minute by all renal corpuscles, meaning both kidneys and all renal corpuscles. Now normally that's 120 to 125 mils per minute, which is a lot, but it can fluctuate, particularly depending on our hydration status, hormones, blood pressure, salt concentrations of blood and interstitial fluids, and the nephron in the filtrate fluid. So we need to talk about the rate that normally occurs and what happens when filtration rate is high and what happens when filtration rate is low. So different glomerular filtration rates will affect the amount of waste that's excreted, meaning waste removed from the blood. Obviously, the higher the filtration rate, the more wastes will be removed. The lower the filtration rate, less wastes will be removed. But these rates also, the glomerular filtration rate of high or low, will also affect reabsorption. And reabsorption is a natural process that happens to, I don't want to say exactly correct for filtration, but fine-tune filtration. So for example, we reabsorb a lot of the water that gets filtered out of the blood. So the higher the filtration rate or the lower the filtration rate, that is going to affect the reabsorption rate, which of course relates to balancing water in the interstitial fluid in the blood and the amount of urine that's generated. Water balance and general glomerular filtration is also tightly regulated to blood volume because of how much water that's reabsorbed and therefore blood pressure. So when we talk about glomerular filtration, we're not just thinking about waste removal, we're also thinking about the volume of blood volume, which you remember is somewhere between 4 to 6 liters, and the blood pressure in the body. So it's easy to take for granted that the kidney filters our blood so that we can eliminate excess waste and excess fluid, but there are some really unique anatomical features of the kidney and the glomerulus in particular that help us, if we learn them, help us appreciate how well adapted it really is for its function. Recall that systemic blood pressure, in the systemic capillaries is low. It's only 10 to 15 millimeters of mercury. But the blood pressure in the glomerular capillaries is 50 to 55 millimeters of mercury. This higher pressure in the glomerular capillaries is due to the different sizes of the afferent and efferent arterioles. If you look at this diagram, you see that the afferent arteriole has a wider diameter than the efferent. So there's a wide inlet for blood, but a narrow outlet for blood to move through the glomerulus. This slows blood flow and it creates a kind of a backup blood pressure. And so there's really high pressure in these particular capillaries, not in systemic capillaries though. When we talk about glomerular filtration, there are 4 main forces that interact to determine what net filtration rate is. It turns out that of these 4 main forces, only three really may play a role or are a factor. But we're going to talk about these 4 pressures so that you know what they are. So there are 2 pressures that are related to fluids. So we call them hydrostatic pressures. The first of these is just plain old blood pressure. And that would be the same as systemic or mean arterial blood pressure. So it'll fluctuate, right? Systolic to diastolic, systolic to diastolic. So we call this hydrostatic pressure of the blood. And then once fluid leaves the capillaries and enters this capsular space, now we have fluid pressure in the capsular space. So that's called hydrostatic pressure of the capsule fluid. I just usually say HP cap, but whatever. So we have pressure of fluid, but then we also have this sort of chemical pressure or solute concentrations that produce a movement, a pressure, a force. And this is called osmotic pressure. And we've talked about osmotic pressure in the blood already. So in the glomerular capillaries, we call this osmotic pressure of the blood. That's what we call it. And when the fluid in the capsular space has a certain osmotic concentration, and so we call it an osmotic pressure of fluid in the capsular space. Now this is the one that really doesn't play a big role. It's very low. In fact, most textbooks say it's 0, but I don't know if that's true, but it has to do with the fact that proteins and blood cells do not escape into that capsular space. So there's a much higher osmotic gradient for water to stay in the blood due to the fact that proteins and red blood cells stay in the bloodstream. So those are the 4 factors, 3 in particular, that contribute to how much filtration actually happens. So the directional forces you can imagine of these 4 pressures are that the hydrostatic pressure of the blood would cause fluid to leave the blood and probably drag some solutes with it. And an osmotic pressure pulls water towards a space. So, we already know this is negligible, this osmotic pressure in the capsule fluid, but if it existed, it would also pull water and fluid into the capsular space. But there are 2 forces that keep fluid in the blood. And one is the hydrostatic pressure of fluid in that capsular space. So there's a little bit exerted backwards. And the greatest factor is that the osmotic pressure in the blood is going to draw water back in. So 2 outward forces that cause fluid to leave the bloodstream and 2 inward forces that cause fluid to stay in the bloodstream. Of these, we are going to see that it's the hydrostatic pressure of the blood and the osmotic pressure of the blood that are going to play the biggest role. But we're going to use some numbers or values and look at why that is so. So looking at some values for the forces that contribute to fluid moving out of the blood, recall that the hydrostatic blood pressure in these glomerular capillaries is 55 millimeters of mercury. And that's due to the difference in diameter between the afferent arteriole and the efferent arteriole. So it's a big out of the blood force, right? The biggest into the blood force is 30 millimeters of mercury. And that's due to the presence of proteins that remain in the blood. And it's about 30 there. 30 millimeters of mercury inward. Now, there's also some pressure that's exerted by the fluid that builds up in the capsular space. and that's about 15 millimeters of mercury. So we can calculate the net pressure for filtration by adding up the positive forces that cause filtration into the capsular space and subtracting the forces from that cause fluid to stay in the blood. So what we get are the outward pressures are about 55 millimeters of mercury, but the inward pressures are about 45 millimeters of mercury. And I just want to mention that this arrow should say 30 on there, not 20. So what we have is a net filtration pressure of 10 millimeters of mercury, which doesn't seem like much. But over time, this causes a lot of fluid, and solutes, of course, to leave the bloodstream. There are several factors that affect this glomerular filtration rate, which, of course, remember again, we're talking about both kidneys and all the glomerular capillaries for all the nephrons, 2.4 million. First of all, it does vary a little bit, the rate. So we just got done talking about the pressure that drives it, but now we're going to talk about the volume of filtrate that's created. So this is a rate. It's how much volume over time. And there's about 105 to 125 mils produced per minute, according to most resources. Some say 100 to 125, whatever. So that's what gives us the 180 liters per minute. But nonetheless, that glomerular filtration rate, producing that much filtrate, 125 millimeters each minute, there's a variation, but it varies within a narrow rate, okay? And it depends, of course, on what is this net filtration pressure. That is it going to stay at this 10 millimeters of mercury? And it assumes that there's a stable surface area for filtration, meaning both kidneys are healthy, all the nephrons are healthy, etc., blood flow's good. And it depends on the permeability of the filtration membrane, which will take into account capillaries, basement membrane, as well as that visceral layer of Bowman's capsule. So we're going to see that what can cause this variation between 105 and 125 mils per minute is going to be fluctuations in net filtration pressure, meaning it won't always be exactly at 10 millimeters of mercury. Primarily, that's because, well, we want it to be able to change with any of our needs, but it also fluctuates slightly based on arterial blood pressure, which also fluctuates. But we don't want filtration pressure to vary as much as blood pressure does. So we're going to see some mechanisms that help keep this tight range of glomerular filtration rate. The reason it's so important to talk about the rate of glomerular filtration is because, of course, it affects excretion of wastes, separating waste from our body fluids, and how much urine we produce. But there's other things too. So let's think about if glomerular filtration is really high for some reason. So you're got a pressure that's higher than 10 millimeters of mercury and a lot of filtrate is being produced. The more filtrate that's produced, then the tubular fluid is going to flow really quickly through the nephron. And if it flows quickly, there's less time for resorption and less contact time with the wall of the nephron. So you are going to get an increase in urine output, which only makes sense. If your filtration rate is high, you should get more urine output. And you'll get more waste excretion with that, but it's at the expense of losing water and ions and anything else that should be reabsorbed. If glomerular filtration is too low, instead, now the fluid is going to flow through the nephron sluggishly, really slow. And now there's a lot of time for reabsorption. And so whatever transport mechanisms there are to, reabsorb things back into the interstitial fluid, even things you don't want, like urea, for example, a nitrogenous compound, then you might actually reabsorb some wastes. And of course, the more you do reabsorption, the less urine you're going to make. So it's important just to make the link right now that high glomerular filtration means you're going to have more urine output, less reabsorption from the nephron back into the body fluids. If GFR or glomerular filtration is too low, then you won't make as much urine, that's true. But wastes might be reabsorbed. You're going to do a lot more reabsorption. So we are able to change, as I mentioned, this glomerular filtration rate within a narrow range. And any change in glomerular filtration rate will affect our blood volume and our blood pressure. And this is related to the reabsorption that we just discussed. So this is just taking the last concept and adding another layer to it. And that is, if glomerular filtration is high, then remember the fluid is moving quickly, and so less water is reabsorbed. If less water is reabsorbed, then your blood volume is going to go down. And if your blood volume goes down, then your blood pressure decreases. So there's a connection between glomerular filtration, the amount of it, and our blood pressure. If glomerular filtration is low, in contrast, remember the fluid moves really sluggish, goes through the nephron really slowly. Now we get more reabsorption, and so our blood volume could possibly increase. Or you get edema, but blood volume will probably increase, and therefore blood pressure will increase. So there's a link between kidney function and blood pressure. So the kidney faces a constant challenge, or both kidneys do. They need a constant, adequate, within normal range, systemic blood volume and blood pressure to filter the blood perfectly. So it needs a constant, non-varying systemic blood pressure. But systemic blood pressure always varies. So that's a constant challenge. And the body needs wastes to be removed, but when the wastes are eliminated, water is lost in the process. Water is precious, and it could decrease our blood volume and lower blood pressure too much. So there's a balance between blood pressure and waste removal, or blood volume and waste removal, right? And there's solutions, regulatory mechanisms, that help maintain adequate pressure in the glomerulus, independent from whatever systemic blood pressure fluctuations are happening. And there are also some mechanisms that override the need for glomerular filtration in order to keep blood volume and blood pressure at a stable level. So you don't have to just use the kidney to regulate how much water is reabsorbed to maintain blood volume and blood pressure. But nonetheless, we're going to look at how the kidney does function to do a lot of that. So the first thing to consider is how glomerular filtration rate can be altered. Before we talk about how the mechanisms work to keep the glomerular filtration pressure, the pressure in the glomerular capillaries, constant, independent from systemic blood pressure, we first just have to know how it can be altered. So recall that the afferent arteriole is wider in diameter than the efferent arteriole. That makes the pressure in the glomerular capillaries quite high. If we were to dilate the afferent arteriole, if it got even wider, that would increase filtration because it would increase blood flow, particularly if the efferent arteriole remains the same diameter. Now we're going to have even more pressure, not just more flow, but we're going to have a higher net filtration pressure. So that will mean more waste removal. So this is useful when you're trying to remove wastes, but there's a low systemic blood pressure. Blood pressure has gone down, but you're still trying to remove wastes. You could dilate the afferent arteriole to keep blood flow maximum through the glomerular capillaries. In contrast, if the afferent arteriole was constricted, narrowed, that's going to decrease filtration. You'll have less blood flow into the glomerular capillaries. And if you have less blood flow, you have less blood pressure. Now, that's useful to prevent water loss with the urine when systemic blood pressure is high. Now, there's something else that you can do that can be done, and we'll examine it a little bit later. Once in a while, not very often, the efferent arteriole diameter can change. Dilation or constriction, and we'll talk about that a little bit later. So it's just some things to review, some things you should understand before you move on, is what effect does a high glomerular filtration rate have on urine production? Meaning, would it cause more urine production or less? And the answer is high. And what effect does low GFR have on waste removal? Well, the lower the glomerular filtration rate generally impedes waste removal. Okay. And that's not just because you produce less urine, but because you have more time for reabsorption of waste products. The third question is, what effect would constriction of the afferent arteriole have on GFR? Well, if you constrict it, you're constricting blood flow, so GFR would go down. It would decrease. The fourth question is, what effect would constriction of the efferent arteriole have on blood volume? And why? Wow, that's really complicated. Okay, so first of all, if you constrict this efferent arteriole, I made up these questions. I don't know why it's so confusing for me. That is, if you constrict it, you make it more narrow. That will raise GFR, at least the pressure, right? It raises glomerular filtration pressure, net pressure. And so more filtrate will be formed. The more filtrate that's formed, the faster the fluid moves. The faster the fluid moves, the less reabsorption occurs, more urine, water's lost, blood volume would go down. So essentially, constricting the efferent arteriole is the same as dilating the afferent arteriole. I hope you like puzzles. Okay, number 5. If a person was dehydrated, would it be more useful to increase or decrease glomerular filtration, and why? Well, it's probably obvious to you now, but decrease glomerular filtration because then less filtrate is formed. You'll make less tubular fluid. It'll move more slowly through the nephron. More reabsorption of water can happen. Less water is lost in the urine, and ultimately it could restore blood volume and blood pressure. If you understood all those things, then I think it's probably okay to move forward. And so now we're going to look at ways that glomerular filtration can be changed, the rate at which it occurs. And there's 3 categories of mechanisms. One category is called autoregulation, and what it means is there's a mechanism within the kidney that self-regulates how fast GFR is. And this is really important for maintaining glomerular filtration when systemic blood pressure fluctuates. You want to keep the GFR sort of steady. But autoregulation can also change the GFR based on sodium chloride concentrations. So it's kind of the kidney can kind of monitor what the tubular fluid components are and change GFR based on that. But the only thing that it really monitors is salt. And I think that's because water balance and blood pressure is so important. But anyway, there's autoregulation. There's also a way for the nervous system to control glomerular filtration rate, and that's through the sympathetic nervous system. The sympathetic nervous system lowers GFR when blood pressure is too low. So when you have low blood pressure, sympathetic nervous system is stimulated. Danger, danger, and now the kidney doesn't get as much blood and it doesn't filter as much. And that's just to maintain blood volume and keep blood pressure up. We'll talk about it in more detail. The third category is hormones. Hormones can change the glomerular filtration rate. And this also happens when blood pressure is low. So both neural and hormonal mechanisms always lower GFR, glomerular filtration, when blood pressure is low, so that water is maintained in order to maintain blood volume. But this autoregulation mechanism that can change GFR, it could increase or decrease glomerular filtration, depending upon what's happening with the systemic blood pressure and what the concentration of sodium is in the tubular fluid. So first we're going to look at how autoregulation occurs. And the purpose, of course, is to ensure waste removal during brief surges and declines in systemic blood pressure. So you don't want too much waste removal and water removal when blood pressure is high, and you don't want a decline in waste removal when blood pressure goes down. So there's 2 mechanisms, and both of these mechanisms can either raise or lower glomerular filtration, and both mechanisms involve the afferent arteriole, either constricting it or dilating it. Now the names of the 2 mechanisms are the myogenic mechanism, and I hope myo makes you think of muscle, so they're going to have some sort of muscular mechanism. And then the second mechanism is called, it's pretty long, is the tubuloglomerular feedback mechanism. I don't know why it has to be so long. This is all about detecting sodium chloride concentration in the tubule. And the tubule will detect it, and that information will be fed back to the glomerulus. So anyway, let's look at them. It'll make more sense once we look at them. So here's the myogenic mechanism. Remember, the whole purpose is to keep filtration rates stable. And it does involve the juxtaglomerular apparatus that we talked about before, or complex, specifically these granular smooth muscle cells that are located in the wall of the afferent arteriole. And just like any smooth muscle cells, when they are stretched, they contract. When they're not stretched, they relax. This is really common in smooth muscle. So let's see how GFR can be maintained. So when systemic blood pressure is high, then before any regulation happens, you're going to have high pressure going into this afferent arteriole, so that the net filtration pressure would increase, generally speaking. But when the systemic blood pressure does go up, that afferent arteriole constricts, and that keeps the filtration rate from going high. So this is how it works. There's high systemic blood pressure, and these juxtaglomerular cells, JG cells, detect a stretch, and they constrict. Once that constriction happens, now there's less blood going into the glomerulus, and GFR slows or lowers. In contrast, if for some reason systemic blood pressure is going down, I know it goes down when we sleep and it goes down when we're dehydrated, but there's other situations, just meditation and things like that. When systemic blood pressure is low, we want to keep glomerular filtration from getting too low. We still need to do some waste removal, right? So in this case, what's going to happen is the afferent arteriole will dilate to keep blood flow adequate and pressure for filtration high enough. So this is what happens when systemic blood pressure goes too low. The JG cells, or the glomerular smooth muscle cells there, juxtaglomerular smooth muscle cells, they don't detect stretch, and so they relax. And if they relax, then the arteriole is dilated, and you get more blood going into the glomerulus, and so that raises the GFR above what, the systemic blood pressure can provide. I want you to realize, though, that this mechanism doesn't alter glomerular filtration rate for the purpose of changing blood volume or changing the pressure. It's all about keeping glomerular filtration stable just for waste removal. That's all. Okay, now we're going to look at the second autoregulatory mechanism, and this is called the Tubuloglomerular feedback mechanism. And again, it's going to involve the juxtaglomerular apparatus, but this time the cells that we're going to be looking at are the cells of the macula densa. They are in that the end of the ascending limb of the loop of Henle. Right, okay, so what do they do? These cells, the macula densa cells, can sense the concentration of sodium chloride in the filtrate. So I'm going to draw again, and I probably should use a different color, but so what you have is filtration, and there's a certain amount of sodium chloride in the tubular filtrate or tubular fluid, and whatever the sodium concentration gradient is, sodium chloride concentration gradient is, at this location will be detected. That's supposed to be a C, and that's supposed to be an L. Sodium chloride right there. That will be detected. Okay, so now what will happen will be that there'll be some vasoconstriction of the afferent arteriole. We'll talk about that in a little bit. But let's just think about why sodium chloride concentration might change or be altered. So if the glomerular filtration rate is too high, like what happens with high blood pressure, okay, the fluid is going to flow very quickly through the nephron, not giving enough time for sodium chloride reabsorption. Now, the reason we need so much sodium chloride to be reabsorbed and not stay in the nephron is all about water following it. So ultimately, this is going to, by reabsorbing sodium chloride, you will reabsorb water. So when glomerular filtration is high, then the fluid's going to move quickly and sodium reabsorption is inadequate. So what happens is the macula densa cells detect that high sodium chloride and stimulate vasoconstricting chemicals, and therefore the JG cells constrict or contract, the afferent arteriole constricts, and now glomerular filtration is lowered. Okay, so that the fluid will slow down. And when the GFR is too low, then the fluid flow is sluggish and you get too much sodium chloride reabsorption. And so what happens is sodium chloride is low in the filtrate, the macula densa cells are inhibited. They don't send a signal for afferent arteriole constriction, so the afferent arteriole dilates and the glomerular filtration rate is raised. So your study questions ask, describe the tubuloglomerular feedback mechanism, specifically whether it can raise or lower glomerular filtration, and it can do either. And then B, the relevance of sodium chloride in the filtrate. If sodium chloride is high, a stimulus is detected, and that's by the macula densa. The role of the macula densa is to detect that high sodium chloride and to release vasoconstricting chemicals. And the role of the juxtaglomerular cells is to cause afferent arteriole constriction. Okay, so that is autoregulation of glomerular filtration. Now we're going to look at neural regulation of glomerular filtration. And this only involves the sympathetic nervous system. And it has a different purpose, less about waste removal, more about maintaining blood volume. So when blood volume is low, too low, you have low blood pressure. And it's important to not filter any fluid out the kidneys. And so blood is redirected from the kidney to vital organs, heart, brain, skeletal muscles, but heart and brain, I guess, would be most important. And so blood flow maintains there, and it's at the expense of filtration. So it's all about maintaining blood pressure and blood flow throughout to vital organs. This is how it works. When blood pressure is low, there are baroreceptors that we studied already in the carotid bodies and the aortic arch. And that signal or that fact is sent via a neuron up to the medulla oblongata. From the medulla oblongata, a sympathetic neuron signal is sent down through various nerves, which we're not going to trace right now, to the JG cells, the juxtaglomerular cells surrounding the afferent arteriole. The sympathetic neuron releases norepinephrine that causes these cells to contract the afferent arteriole, and so now that lowers GFR. So low blood pressure, and you will lower the glomerular filtration. This is going to slow how fast fluid flows through the nephron, so that there's more time for reabsorption of waters and salts, because water is going to follow the salt. So your study question asks, describe how the sympathetic nervous system affects GFR. It lowers it. How does it affect urine output? It lowers it. How does it affect blood volume? It raises it. And how does it affect blood pressure? It raises it. Okay, bear with me, we're almost done. The only thing we have left are hormones. So the glomerular filtration rate can also be controlled with hormones, or at least slightly regulated within its narrow range by hormones. And low blood pressure is going to stimulate these same JG cells that we've been talking about to release renin. And the low blood pressure causes them to release renin because they're not being stretched. The other thing is the sympathetic nervous system also causes them to release renin. But the important thing is these cells make renin when blood pressure is low. So when blood pressure is low and renin is produced, this is ultimately going to increase glomerular filtration rate, which is the opposite of what we just saw, right? And it does so in a different way. It does so by constricting the efferent arteriole. The other thing it does is it raises systemic blood pressure, renin does, by activating downstream many other hormones, particularly angiotensin. This is a potent vasoconstrictor, and so your blood pressure will go up. So let's look at how renin works on the glomerular filtration rate and the reabsorption process. So constriction of this efferent arteriole right here, if you made that more narrow, that would cause backup pressure in the capsule. And so you would have higher net filtration pressure, and you would get more fluid, more filtrate formed. So you would actually increase the rate of filtrate formation. The other thing that renin does by constricting this efferent arteriole is that, I'll call it downstream from that location, these blood vessels, these capillaries, the paratubular capillaries, are going to be experiencing lower blood pressure. It's when we talk about vasoconstriction, it's always hard to know, is that increasing or decreasing the pressure? Well, it depends if you're upstream or downstream. So when you constrict this vessel right here, you're causing higher upstream pressure but lower downstream pressure. Okay, so the lower pressure in the paratubular capillaries promotes more reabsorption into them. So although you've increased the production of filtration or filtrate, you're also increasing reabsorption back into the blood. So it's a really cool mechanism because you're going to get rid of wastes, but you're going to maintain some water. There's a lot of other things that renin does. In a stepwise process, it activates Angiotensin II, which is really the vasoconstrictor. So what renin does is converts angiotensinogen into angiotensin I. Now angiotensin I is floating around in the blood, and it goes through the pulmonary capillaries where it's exposed to an enzyme called ACE, and that converts angiotensin I into angiotensin II. So really, what has renin done? All it's done is produce angiotensin II, okay? But angiotensin II has an effect on blood pressure, not only through vasoconstriction, but also by affecting glomerular filtration. So here are the functions of angiotensin II. widespread vasoconstriction. That would include in the afferent arteriole. So it would increase net filtration pressure. But another thing that angiotensin does is it increases nephron's reabsorption of sodium and water. It does so directly by acting on those two convoluted tubules, making them more permeable to sodium and water so that there's more reabsorption. What is that going to do? It's going to raise blood pressure, which is the same thing as vasoconstriction does. But it also, angiotensin also indirectly affects sodium and water reabsorption by causing the release of antidiuretic hormone and causing the release of aldosterone by the adrenal cortex. But all three of these things result in more sodium and water being reabsorbed by the kidney with the purpose of raising blood pressure. So here's a diagram that shows all the things that angiotensin can do, sort of, almost all. Vasoconstriction, which will increase glomerular filtration, but in addition, angiotensin II acts on the kidney to promote reabsorption and on the adrenal cortex to make aldosterone, which promotes reabsorption. It also acts on the hypothalamus to cause thirst, drinking, and produce posterior pituitary to make antidiuretic hormone. So there's just tons and tons of things that angiotensin does to raise blood pressure, but we still get, generally speaking, wastes removed. So this basically is a slide that says everything I just said. You would think that angiotensin II, since we have widespread vasoconstriction, including the afferent arteriole, you would think that would actually lower GFR. And that's true initially. Initially, glomerular filtration rate is lowered. And that is to prevent too much urine formation and preserve water so that you're raising blood volume and systemic blood pressure. However, as you slightly get a raise in systemic blood pressure, then you're going to get more of a raise in glomerular filtration. And that happens because this tubular fluid has slowed because of the low glomerular filtration. So more water has been reabsorbed, and that's how that blood pressure was raised. So it functions to maintain adequate blood pressure and keep waste removal continuous. I thought I'd mention how antidiuretic hormone works. Specifically, if you recall, it comes from the posterior pituitary. It acts on the cells lining the collecting ducts, making them more porous to water. ADH stimulates the synthesis of aquaporins in these collecting ducts, so more water can be reabsorbed. And we'll discuss that again when we get further along in the nephron. Aldosterone, which is made by the adrenal cortex, generally works on the distal convoluted tubule, but some say also the collecting duct. But what it does is it stimulates sodium reabsorption at the expense of potassium secretion. So potassium is going to go into the urine. Now the important part, though, is that sodium reabsorption leads to water reabsorption, because remember, water always follows sodium. That was a lot of information. Thank you for listening so long. And our next topic will be on processes of tubular reabsorption and secretion. Thank you.